Antibody-dependent enhancement is a mechanism to explain why secondary infections of dengue virus is much more severe than the first. It is important to recognize that immunity against one serotype of dengue does not automatically mean immunity against the other three serotypes. When you first get infected with dengue virus, circulating macrophages that have MHC receptors engulfs it. The macrophage processes the virus and presents the antigen on its surface. Normally, circulating cytotoxic T cells ignore healthy cells and pathogens, but it recognizes the MHC antigen complex. When the cytotoxic T cell binds to the MHC antigen complex, it gets primed. Priming allows the cytotoxic T cell to recognize a specific antigen to target. Next, the T cell undergoes division into two subgroups, effector cells and memory cells. Effector cells target infected body cells, while memory cells make antibodies that target the pathogen. When you get infected by the same serotype again, the antibodies that were made during the initial infection would bind to the virus. The binding signals macrophages to engulf the complex, thus neutralizing it. When you get infected with the new serotype, the antibody from the initial infection binds to the virus. Although the antibody is capable of binding, the new serotype differs from the first, so the antibody can't neutralize it. When a macrophage comes by, it does not recognize the complex as being foreign, so no phagocytosis occurs. Instead, the antibody-virus complex binds to the Fc receptors of cells such as monocytes. When this complex binds to the cell, the virus gets ingested. Once inside, the virus begins to replicate. When the virus is ready, it gets released from the host cell. Essentially, the non-neutralizing antibody acts as a piggyback for the virus to expand its infectivity range. Initially, the new serotype would only be able to infect a limited amount of cells that contain a correct attachment molecule, but with the help of the antibody, it can infect a larger amount of cells.